Okay, let's get started. Today's study tip says, talk out loud to yourself as you explain a topic. Speak for at least 2 minutes about what you know. So I had a student years ago who was trying to become a nurse. She was an accountant. I remember she worked full-time, so she always came to my night class dressed up in her accountant outfit. And she had 2 kids, and she told me she was doing well in the class, so I asked her how she studied, and she said she put her kids to bed, and she'd go in her room, shut the door, stand in front of her mirrors, which were the closet doors, and she would talk out loud, just like this study tip. So it really worked for her. I could not do this, no way. But she would just say it all over, and then of course you can't maybe do 2 minutes, but 15 seconds, and then review some notes and talk out loud. Hey, whatever works. Okay, so I give all these study tips just so you can hear what other people are doing. So today we do Chapter 7, Part 2 on neurophysiology. So we set it up last time, we got the action potential. Now we need to do the parts of the synapse. So this is a reminder of the anatomy. The top, the brown part here is coming from what we call the presynaptic neuron. This is the dendrites, the dendrites are the yellow part of the postsynaptic cell, and the axon terminus of the presynaptic axon are the brown part. Okay, so this is where they're talking to each other, and the part where they talk to each other is called a synapse. So it's a neuron talking to a neuron. There's also a synapse where a neuron talks to a muscle or something else, but today we're going to focus on neuron talking to neuron. This is called the cell body, the soma. Remember, the axon hillock. This is the axon hillock, called the axon initial segment here. This is the most sensitive region of the dendrite. What that means is that it reaches threshold more easily. So remember, we're trying to go from -70 to -55, and to reach threshold here might be easier. So what does easier mean? -50 or -60? So if it's easier to reach threshold, would you have a threshold of -50 or -60? Like -60, right? You wouldn't have as far to go to reach threshold from rest. And then an action potential is created. And by the way, one neuron like this would have maybe 100 talking to it. And they all are talking, or some are not talking, and some are. And we'll talk about how it knows what to do based on who's talking the loudest. Okay? Remember the zipping, the saltatory conduction down the axon. There's the nodes of Ranvier, the myelin, all the way down. And then the neurotransmitters are released. Okay? So the synapse is a communication between 2 neurons. The first neuron is the presynaptic neuron. The book calls it, remember, the terminal bouton? I don't know. Like it's French? I don't know. I don't think so. Synaptic knob, the axon terminus is what I call it usually. Synaptic knob is good because it looks like a doorknob, right? Reaches out to many more neurons. Whoops, wrong way. And the second neuron is the postsynaptic neuron. It's the most sensitive region in the the most sensitive region, really. The most sensitive region is the axon hillock. And it has a threshold that's easy to reach. So we talked about that. So this is how your book explains it. It shows the action potential coming down the synaptic knob is here. This here, remember, synaptic cleft. And the bottom is the postsynaptic cell. When the action potential reaches the synaptic knob, calcium channels open. So step 1, action potentials reach the axon terminals. Step 2, voltage gated calcium ion channels open. And calcium comes in. So that reminds you, if it comes in, where is calcium the highest? On the outside or the inside? Highest on the outside. So it comes rushing in. It binds to a sensor protein in the cytoplasm. I'll tell you the name of that protein on the next slide. And then they come over, and the synaptic vesicles are here at the end. And the calcium protein complex stimulates the fusion and exocytosis, we know this, right, of the neurotransmitters. So these vesicles are kind of stuck here with this protein called the snare complex. Okay, you don't have to know what snare stands for. But when the calcium and protein get together, it causes fusion of the vesicle with the plasma membrane, and then it releases the neurotransmitter into the synaptic cleft. Okay, so I'll show you a video of this next so you can kind of see how this goes. Okay, this is the animation of the chemical synapse. Let me read it to you. Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft, bind to the receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced. So this is easy. I mean, you know more details, and I expect you to know more because you know the real parts of the synapse, the action potential arriving, how the release of the neurotransmitter, but I wanted you to kind of see how this works. But I expect you to know far more detail, okay? Okay, so here's what I need you to know. The steps of a synapse. 1. Action potential reaches the knob, synaptic knob, right? The voltage-gated calcium ion channels open. Step 3, calcium influx. 4, calcium binds to synaptotagmin. Try saying synaptotagmin. Yeah, that's a good one. Synapto for the synapse, right? And then the complex releases the snare proteins that hold the synaptic vesicles in place. And it keeps going. Step 6, the vesicles with neurotransmitters fuse with the plasma membrane. Remember, that's called exocytosis. The neurotransmitters are released into the synaptic cleft, and the neurotransmitters bind to receptors on the postsynaptic neuron. Okay, get this in your memorizing mode. Okay, how are you going to do this? Practice, talk out loud. What are you going to do? Fun fact, even if we lose 10,000 brain cells a day from the time we are born, we have started with so many that the total number lost by the age of 80 would be less than 3%. So I'll tell you what, as I get older, I don't want to lose any of them. So I know you think 80 is so far away, only 3%, but I'd like to keep all of them. But I'm sure you know a lot of 80-year-olds don't have everything working as well. And I hope we all do. Next, ligand-operated channels. Now, we're going to talk about 3 different ways that the neurotransmitter binds to the postsynaptic cell. The first one is called the ligand-operated channel. This one does use the example of acetylcholine. So on the left side, you see that right there. This crosses the cleft, binds to the channels. So here you can see on the right, this channel is closed. One acetylcholine has stuck, the other one has not. When it does, it opens a channel. This channel happens to open sodium and potassium at the same time. These are called nicotinic acetylcholine receptors, and they are nicotinic as in the nicotine that you know from cigarettes. So these, especially on the heart, let's just do the heart. On the heart, what does nicotine do to your heart rate? If you smoke, what happens to your heart rate? It causes it to increase, right? So when acetylcholine binds to its receptors, nicotine does a very similar thing to receptors, causing the heart rate to go up, and acetylcholine causes the heart rate to go up, which is good if you're running, right? If you're taking nicotine, it's not, because you're smoking while sitting and your heart rate goes fast, and of course that's bad. But if you're running and you're making acetylcholine, it's binding to receptors, it's making your muscles go, making your heart go. Perfect. That's how it's supposed to work. So these are the, oops, still can't go the right way, I'm sorry. I need my coffee. The ligand-operated channels are the first one. Easy, the acetylcholine, which we write as capital A, capital C, lowercase h, for short. You, of course, on the test will write the full word, acetylcholine, and then put ACH in parentheses for me. ACH crosses the cleft, binds directly to the ion channel. The ion channel opens, and then I just wanted you to notice that 2 acetylcholines are required to open the channel. Okay, now we get a little harder. In this story, it's called a G protein operated channel. Acetylcholine comes this time and binds to a receptor, but the receptor is not a channel. Okay, this receptor instead stimulates some proteins here called G proteins. I don't know why they're called G proteins. probably named after somebody. You know how that goes. Then when these are triggered, there's three subunits. Can you see that in the picture? There's alpha, a beta, and a gamma. The beta and gamma dissociate, right, and come over to a channel, in this case, potassium channel. I don't need you to know which subunits dissociate. Okay, I just need you to know that, as it says here, the G protein subunit dissociates, and then it binds to, in this case, a potassium channel, causing it to open. And which way will potassium go, in or out? Potassium goes out, right, because it's highest on the inside. By the way, potassium leaving, would that make a depolarization or a hyperpolarization? If positive charges leave, the inside becomes more negative, right? Is that a depolarization or a hyperpolarization? That's a hyperpolarization, right? So this gets you further away from threshold. So in this case, this neuron would be is told to not turn on, okay? Which happens also, right? So we turn things on and we turn things off. Okay, so here's the steps of the G protein operated channel. Acetylcholine crosses the cleft. Acetylcholine binds to the protein receptor. G proteins are activated and dissociate, and the subunits open an ion channel. And I'm being vague, okay, because we could say potassium and potassium goes out, but we're just going to be vague. Depends on what organ or what target cell we're talking about. Then it gets harder. By the way, all of this pays off, because when we do the endocrine system, you're going to have this, you're going to see this again. So to memorize it now is going to pay off later. This gets a little more difficult, lots more steps. First thing, the acetylcholine is not this part of this story. Instead, we have a neurotransmitter called norepinephrine. Have you heard of it? It's like epinephrine, right? So it's going to make your heart rate go up and all that stuff. Here it binds to its receptor. Similar stories. Remember, this is a protein with a perfect lock and key fit. G proteins, again, dissociate. In this case, it's the alpha, but I don't need you to know that. Just know that it dissociates. And then that triggers a protein called adenylate cyclase. ACE means enzyme, right? And this is going to take ATP and make it into cyclic AMP. It looks like the word cycle, but we say cyclic AMP. M, it stands for mono. So how many phosphates are on this? 1 versus ATP has 3. So we've lost 2 to make AMP. The cyclic AMP takes a protein kinase, again an ACE, which the job of a protein kinase is to phosphorylate something. Okay, so this is an inactive version of the protein kinase. It's an enzyme. Becomes active with cyclic AMP. That then phosphorylates proteins, which might be an ion channel that's phosphorylated, and when that happens, it opens. And we're being vague again, not showing what's going through. Okay, so let's write down the steps how I need you to know, okay? So the second messenger system is called norepinephrine, and the example is norepinephrine crossing the cleft, binds to protein receptors. G proteins are activated and dissociate. Subunits signal adenylate cyclase. Then adenylate cyclase, which I abbreviated as AC, you won't see that in your book, but I was running out of space on my slide, okay? Adenylate cyclase converts ATP into cyclic AMP. And a lot of people read it as little c, big AMP. We don't usually say camp, though. CAMP activates the protein kinase. The kinase phosphorylates an ion channel, so it will open. Okay, so that's 4 step things we did already. We did the steps of the synapse, did the second messenger system, right, the G protein, and the ligand-operated channels. Now the signal will continue to go until it's halted, and to stop the synapse, you can do a couple things. The neurotransmitter says will continue to go. The enzymes in the synaptic cleft will degrade the neurotransmitter. So that's one way to make it stop, right? You have enzymes that chop up the neurotransmitter. Or, second thing, they are recycled back into, it just kills me to say this word, so I'll say the axon terminus, okay, where they came from. So you can recycle them, take them back. So here's a picture of how this would look. So here we have the, at the top here is the presynaptic axon, synaptic cleft. Bottom is the postsynaptic cell. This picture is showing acetylcholine coming from the synaptic vesicles, binding to its receptors. So the receptors are these are going to be a G protein story here. You see how the orange one? But when these purple things are there, the purple thing is an enzyme in the membrane called acetylcholine esterase. Try saying acetylcholine esterase. And it takes the acetylcholine, chops it into two pieces here, acetate and choline. And now this no longer works to bind to the G protein receptor. Okay, so since it can't bind, then we no longer have opening of sodium channels, so we no longer have an action potential and no longer have a synapse. The other choice besides chopping is to recycle. So we've learned the word exocytosis, right, which is the release of the neurotransmitters. What's the opposite of exocytosis? Endocytosis. So bringing them back up into the presynaptic cell would be like the recycling kind of thing. So some people who suffer from depression have a lack of serotonin in their synapses. Have you heard of that? And they have, pretend the green ones are serotonin. They want more serotonin in their synapses to prevent the depression. And so one of the drugs that we give is a serotonin reuptake inhibitor, SRI. They're called SSRI. I forgot what the first S stands for. But those serotonin reuptake inhibitors are drugs that actually prevent this from happening, this recycling, so that instead the dots here stay in the synapse instead of going back to be recycled. Okay? Lots to learn here in this. Next. Table 7.7 has a category of neurotransmitters. I'd like you to know the basic categories and an example of each one. Okay, so look in your book for the examples. I'll just tell you basically what it says. It says amines are all derived from the amino acid tryptophan. So remember when we were talking about proteins, talk about amino acids making up proteins. There's one called tryptophan. I don't know if I mentioned it, but that's the one high levels found in turkey. So it's one of the things supposedly that makes you sleepy at Thanksgiving after you eat. I think it's just because we're all tired and exhausted and we're full, right? Tryptophan is an amino acid that is used by your body to make amine neurotransmitters. Catecholamines, very similar, derived from the amino acid tyrosine. Next, other amino acids, I won't go into the names of them, but there's a very common neurotransmitter in the brain called GABA, G-A-B-A. We do say GABA. And polypeptides can be neurotransmitters. Remember, polypeptides are short little proteins. We also call them hormones. And even gases can be dissolved in your blood, and they're supposed to be there, and they act as neurotransmitters. Okay, so you should know the categories and an example of each. Okay, excitation of neurons. So the excitatory postsynaptic potential, look at this word, excitatory postsynaptic potential, called an EPSP. You know I like to make this stuff up. So I didn't even make this up. This could have been a word I made up. It's due to the opening of sodium channels, and it depolarizes the membrane when there's an excitatory postsynaptic potential. Okay, so right, you open sodium channels and sodium comes rushing in and you get depolarization. But you need more than one EPSP to reach threshold in the postsynaptic neuron. Okay, so if you have 100 cells talking to one, if one of the 100 is talking and saying, okay, I'm going to excite the second cell and open sodium channels, it's not going to be enough to reach threshold. So this picture is showing kind of how that would work. So on the left side we see the millivolts, minus 70, minus 55, plus 30. Here's your threshold. Here we have a picture at the same time that they have a graph. Picture of cell 1, cell 2, both talking to the postsynaptic cell. Okay. If cell 1, it says release of neurotransmitters from neuron 1 only. Neuron 1 would release neurotransmitters and open sodium channels, but look, that's not enough to reach threshold. But if both neurons do that, you get enough sodium channels open, enough sodium coming in to reach threshold, and then you get an action potential. So this says release of neurotransmitters from neurons 1 and 2. So spatial summation, this is called spatial summation. Sum because they're sum together, right? Addition. And spatial because they're separated from each other. Okay, you could also have one cell give a really fast boom boom, right? Really fast signal of neurotransmitters. That also could open up sodium channels fast enough to reach threshold. Okay, so types of summation. The temporal summation was what I was just talking about. Several EPSPs from the same neuron. So excites really fast so that you build up the sodium enough to reach threshold. And then the spatial summation, which is what the picture is that I just showed you, that's an EPSP from different neurons, but enough to reach the threshold. Okay? There's also a way to inhibit neurons, right? We saw this. This is the inhibitory postsynaptic potential. And we saw this in the picture, the purple protein, remember, was potassium channels, right? It could also be not just potassium leaving, but chloride channels opening, because remember, where is Cl the highest? On the inside or the outside? On the outside, right? So when you open Cl minus channels, negative comes in, and that hyperpolarizes the membrane. So when you send an IPSP from presynaptic to postsynaptic cell, threshold is not reached, and an IPSP from one neuron plus an EPSP from another results in nothing, right? To inhibit and to excite. And I don't know if your book still calls it this, but it used to call it an EPSP IPSP cancellation. I swear. I don't even make this stuff up. Someone else does. Okay, so you got the basics. You know all of the neurophysiology that you need now for this class. Start memorizing. We're done.